All right, today we are taking a look at 8.3. We're going to take a look at some cell cycle checkpoints. So 8.3 cell cycle checkpoints, kind of a continuation of what we were taking a look at yesterday with the cell cycle. So throughout the cell cycle, there are a series of checkpoints that are controlling that the cell grows and then it goes through the cell cycle and eventually divides. And we want these checkpoints, and we want these checkpoints to maintain control. There's actually a term, unfortunately, we're probably all too well aware of it. There's a term for when these checkpoints fail, for when the cell goes through cell cycle without any constraints. And that term is cancer. It's that terrible disease we've all probably been affected by one way or another. Cancer is really a loss of the cell's ability, of the body's ability to control which cells grow and divide. So when cells kind of go out of control and start growing and dividing, they end up forming a tumor, and the end result is cancer. So these cell cycle checkpoints are really, really important. There's going to be 3 of them that we'll discuss today. So the first of those checkpoints is at the end of G1. You guys remember from yesterday, G1 occurs just after the cells have divided. It's a growth phase where the cell's growing, it's replenishing its organelles. And before we go any further, we have to have a checkpoint. At that checkpoint, we're going to ask these questions. Is there a need for the cell to divide? Are there room, food, resources for new cells? If it's a crowded area or there's not enough food or resources, the cells won't divide. They'll never get turned on. They'll never go through the checkpoint. And the cells won't divide. And again, that's a good thing. When the cells divide, you're going to end up with new cells, and that's more mouths to feed. So if there isn't food for them, there isn't room for them to divide, we don't want them to. We want to stop them at the end of G1. If you think about it, following G1 is S phase. And during S phase, that's where the DNA gets duplicated. We have a lot of DNA. It's going to take quite a bit of time, quite a bit of energy, and tons of resources to duplicate our DNA. If there's no reason for the cell to divide, then there's no reason for the cell to duplicate its DNA. So it's really important that we determine, hey, we need to divide. Before we do that, we have to copy our DNA. Really important to do that at the end of G1 before we enter S phase. If they don't pass this G1 checkpoint, if they can't answer affirmatively to these questions, cells move into what we call a resting phase known as G0. And G0 is a fine place. I'd wager a bet most of my cells currently are in G0. They're just living their normal everyday life, doing the task assigned to them and doing it well without growing and dividing. There isn't always a need for it. So cells will oftentimes spend a large portion of their life in G0, in this rest phase, not dividing. Often, a lot of cells can go into G0 and can come back out of it kind of at their will. Not necessarily at their will, but if that cell checkpoint gets passed, if they go into G0 and sit there for a while, and then all of a sudden, hey, the cell next to you got damaged, you need to divide and replace it, you can come out of G0 and go back into the cell cycle. So that's the first checkpoint, the end of G1. The next checkpoint is the G2 checkpoint. I'll let you go ahead and take a guess as to where that one occurs. So by the end of G2, we've duplicated our DNA. We have grown larger as a cell. We are preparing to divide. So the questions asked here at the end of G2. Is the cell ready to divide? Has it made enough organelles that when it splits in half, you're going to have two new daughter organelles? Is it big enough? Is it large enough? So that's the first question. Second question, and this one's super important. Did the DNA copy correctly? So remember, by G2, we've been through S phase. We've duplicated our chromosomes. We've copied our DNA. We need to make sure that this happened correctly. First of all, we need to make sure we have enough DNA. Did we copy every single chromosome entirely? As humans, we have 46 chromosomes. What if you only copied 45 of them? You didn't copy the 46th. When you make two new cells, one of those cells is going to be shorted a chromosome. And it's not something you can fix after the point. Once those cells divide, it's too late. You can't go back in and add DNA after the fact. So we have to make sure we are ready to go. We have to make sure our DNA copied entirely before we go ahead and enter mitosis, where we're going to split that DNA up. In addition to make sure we got all of our DNA copied, we also have to make sure we did it correctly. Mistakes in the DNA are called mutations, and we'd rather not have many of those. Every once in a while, they do slip through, but we're going to do our best to catch them as much as we can to make sure that our A's paired with our T's and our C's paired with our G's, and we're going to check for that here at the G2 checkpoint. Now, a little bit different than the G1 checkpoint. G1, we went into G0, just resting, and the cell kind of lives its life. If you don't pass the G2 checkpoint, yes, you can kind of go into an arrest, but during that time, we're going to try to fix these problems. We're going to try to make sure we are ready to divide. We're going to try to make sure that we can fix our DNA. We're not just going to hang out at the end of G2 for an entire life. We've got a whole bunch of DNA. We're ready to go. Let's get ourselves ready to divide. All right, our third checkpoint is at the metaphase-anaphase transition. So we haven't yet got to mitosis. We haven't talked about it just yet. So this will make more sense tomorrow when we do get to mitosis. But for right now, as we transition from metaphase to anaphase, we have our third checkpoint. And what we do there, and this will make more sense tomorrow, that third checkpoint between metaphase and anaphase, we're going to check and make sure that the spindle fibers are correctly attached to their chromosomes. Again, we'll see more about that tomorrow when we get into mitosis, but that's a very important thing to do as well. Those are the three big checkpoints we're going to talk about. Well, let's spend a minute here talking about how those checkpoints are regulated. These checkpoints are regulated by a couple of proteins known as cyclin-dependent kinases. So we've got kinases and cyclins, these two proteins that work together, they're cooperative. In fact, technically, kinase is kind of the big enzyme, the big protein, and cyclin is just a little cofactor that kind of fits in the kinase and makes them go. But both of them are there, and both of them are what keep the cell cycle running. So together, and again, this is key, both cyclins and kinases have to be present in order to work. So together, they act as kind of like a key, and they start the cellular machinery that drives cell growth and division. So they're what keeps you going through the cell cycle. But if they're both not there, the cycle shuts down. The cell stops down, it stops moving forward. My analogy, kind of a goofy one, but my analogy, so we have kinases, and we have cyclins. And together, they're going to work really well. Individually, neither one of them works. You can turn this one, but you can't fit it into a lock. This one fits into the lock, but you can't turn it. So together they have to both be present. They have to both be present. They're going to fit together. This is our CDK complex. We have our cyclin and our cyclin-dependent kinase linked together. And when they're both there, we're ready to turn on the cell cycle. So that's my little analogy there with these two proteins. They both need to be there to move that cell cycle forward. It provides for a system of checks and balances. It allows part of our cell to be like, hey, we're ready. Let's go. Let's proceed. And the other part to say, wait a minute, wait a minute, wait a minute. We're not ready. We're not ready. And so both parts have to be ready in order to proceed. And that's the idea with our cyclin-dependent kinases that are driving the cell machinery. All right, I hope we see you back here tomorrow. We'll get into mitosis tomorrow. See you then.